Chapter 13b, Brains. In the first part of this rather lengthy lecture, I'll cover more about meningitis, and we'll talk about why spinal taps can be used to diagnose it, and why treatment with antibiotics runs into problems. Next up, in the classroom, I would normally illustrate the function of the cerebellum by trying to play this online video game called QWOP. I can't do that and record this video at the same time, so take a look for this game, and it should illustrate to you why your cerebrum is really bad at doing things that your cerebellum does without much effort. We'll also talk about why the brain is hollow, and why the forebrain is not only located in the front, but the sides and the back of the head as well. To do all of that, we'll start with the basic overview of brain regions. We'll talk about how the brain develops, move on to discuss the ventricles and the meninges. These should be review. And then we'll start discussing individual parts of the brain, starting closest to the spinal cord with the hindbrain, and moving our way up to the midbrain and cerebellum. The 6 major regions of the brain include the cerebrum, cerebellum, the diencephalon, the mesencephalon, the pons, and the medulla oblongata. We'll be covering these one at a time. The cerebrum is the largest region of the brain. It's the wrinkly part that's most visible from the surface. Each bump is called a gyrus. Each little dip in between the bumps are called sulcuses, and there's some really large gaps called fissures. The cerebellum is also wrinkly and fairly large. The diencephalon is a group of structures that are deep to the cerebrum. I would like to focus on their individual parts and their functions rather than talk about this unit as a whole. However, altogether, the diencephalon is responsible for a number of emotional responses. The thalamus is a pair of oblong structures on the left and right sides. Below that is the hypothalamus. Sometimes we will even include the pituitary gland as part of the diencephalon. However, the anterior pituitary is epithelial tissue and not neural tissue at all. Moving in a rostral direction, we'll next come to the brain stem, which includes the midbrain, pons, and medulla oblongata. These regions of the brain are responsible for very basic survival functions. The midbrain, or the mesencephalon if you're not into the whole brevity thing, is a rather difficult structure to identify in lab. I usually identify the easier structures surrounding it, and then find the midbrain by process of elimination. The pons is one of those easier structures to identify. It's an oblong bit shaped a little bit like a small lime. Lastly, the medulla oblongata looks a lot like the spinal cord. The major difference here is that this is located inside the skull. Once this part exits the foramen magnum, we would then start calling it the spinal cord. Those are the basic regions of the brain. We'll be working from the hindbrain upwards. But first, let's discuss how the brain develops. Back when you were an embryo, a little region of skin on your dorsal surface grew thicker and eventually rolled up into a tube that we call the neural tube. This is going to give rise to both the brain and the spinal cord. Here are some pictures of the neural tube as it is folding in this early stage embryo. You will notice that this tube is hollow. Most of the tube stays tubular in shape and becomes the spinal cord. But up in the head, this tube begins to grow rapidly in 3 major bulges. And if you were a simpler organism, these bulges would stay organized thusly: forebrain up front, midbrain in the middle, and hindbrain in the back, and everything would be nice and logical. But being a human being, your brain had to overgrow and things got weird. Specifically, it's the forebrain that overgrows. And because it's limited by the shape of the skull, it's going to be pushed towards the back and then curl around to the sides as it continues to grow larger. This is going to be the reason why we've got forebrain in the front, the back, and the sides. Here's a different picture from our textbook that shows the growth of the fore, mid, and hindbrain in very early stages of development. If we add to that the limit in shape and size that's imposed on it by the skull, here we can see the forebrain growing in its particular fashion. It can't keep growing forwards, so it has to grow backwards and then off to the sides. To review, our 6 major regions of the brain are the cerebrum, the diencephalon, the midbrain, cerebellum, pons, and medulla oblongata. And these develop from the forebrain, the midbrain, and the hindbrain, which all in turn come from the anterior portion of the neural tube. These are the names that I'm going to be testing you on. But first, let's cover some very important cells that started off as a part of the neural tube, but then migrated away to form some very interesting and important structures. These are called the neural crest cells, and some of them leave to go become part of the skin, forming melanocytes. Others form things that look more like the nervous system, like ganglia and the adrenal medullae. But some of these neural crest cells become connective tissues, including some very important cartilages up in the neck. And now it's story time. Here's a picture that I took of some neural crest cells migrating away from the central nervous system. This was part of a large project. One of my coworkers had made a transgenic fish, and specific cells glue in the dark. It was my job to take pictures of them, and hopefully to make a movie of the developing nervous system over the course of several days. My first few attempts at this failed. I stayed at school for 3 days straight, taking one photo under the microscope every 2 hours, only getting small breaks in between to nap and eat and whatnot. But the movie failed because the fluorescent protein faded over the course of a day or two. I tried a third time, and that as well failed. And so I have here this photo that I'm going to jam into my lectures year after year just to get some use out of it, because I spent a lot of time at school getting very little sleep. Next, we can talk about the ventricles of the brain. Because the brain and spinal cord started off as a tube, the hollow portion winds up persisting throughout adulthood, forming the central canal of the spinal cord and the ventricles in the brain. The ventricles take on a more complicated shape than the central canal of the spinal cord, and that's because of the way the forebrain grows. The ventricles started off 1, 2, 3, 4 in order from forebrain to hindbrain, but as the forebrain grew towards the back and off to the sides, the first and second ventricle fused together and then split into a pair of lateral ventricles. The third ventricle and the fourth ventricle still have relatively straightforward names based on their location. Inside of the ventricles and central canal is cerebrospinal fluid, which is one of several mechanisms that help protect the brain physically. We also have a biochemical protection for the brain known as the blood-brain barrier, and these are the things that we need to discuss next. Let's start with the meninges. These are the same structures that we covered in the previous chapter around the spinal cord. We have a thin layer of pia mater, a layer of arachnoid mater, beneath which we have cerebrospinal fluid surrounding the brain, and then the most superficial layer and toughest, the dura mater. There are two major differences between the dura mater surrounding the brain and the dura mater surrounding the spinal cord. The dura mater in the brain has a hollow center in which are located dural sinuses. Fluid will collect here and drain back down towards the heart. Furthermore, the dura mater folds in between the hemispheres of the cerebrum and cerebellum, and these folds get specific names. The falx cerebri is a fold of dura mater between the two hemispheres of the cerebrum. The falx cerebelli is between the two hemispheres of the cerebellum, and the tentorum cerebellum rests above the cerebellum like a tent, separating it from the cerebrum. Cerebrospinal fluid drains into the dural sinuses once it has been used up, and the dural sinuses can carry it back to the heart to rejoin the circulatory system. One major function of cerebrospinal fluid, which is located in the subarachnoid space surrounding the brain, as well as the ventricles inside of the brain, is to cushion and support the brain. I have been told that the consistency of living brain matter is very similar to soft tofu, and just like soft tofu, you need to keep it suspended in water, otherwise it crushes itself under its own weight. Cerebrospinal fluid can also provide nutrients to the inner and outer surfaces of the brain. Cerebrospinal fluid is made in specialized structures called the choroid plexuses. These are groups of ependymal cells that make large amounts of cerebrospinal fluid, about half a liter every day. They make it by filtering out water and nutrients from the bloodstream. After being made in the choroid plexus, cerebrospinal fluid will circulate through the center and around the outside of the brain, traveling through the ventricles and the central canal, down and around and back up around the subarachnoid space of both the brain and the spine. Once the cerebrospinal fluid has lost most of its nutrients and built up wastes from the brain, it needs to drain into the dural sinuses, and it does so at specialized structures called the arachnoid granulations. It's important that the entire brain, not just the inner and outer surfaces, receive nutrients. Neural tissue cannot store any of its own nutrients, the way that skeletal muscle and adipose tissue can. So it requires a constant supply, and this is carried out through an extensive network of blood vessels throughout the brain. If these blood vessels become occluded, a portion of the brain becomes starved for nutrients, like glucose and oxygen, and very quickly becomes damaged and can even die. The old-fashioned name for this was a stroke. More commonly these days, you might call it a cerebrovascular accident. While that would of course be very bad for your patient, this is how we know what we know about the human brain. We found people who have survived such damage to their brain and asked what sort of changes have occurred since then. And based on those changes, we conclude that that part of the brain must have been responsible for doing whatever it is that is now different. This is, of course, a very crude way of learning about the human body. It would be kind of silly to go into an auto mechanics class and have the instructor remove a part and then ask what's wrong with the car now. But that's how we do it. Next is the blood-brain barrier. When I learned about the blood-brain barrier, I asked my instructor where it was located, and he said somewhere in the neck, and that was entirely wrong. The blood-brain barrier is located throughout the entire brain. It's not a single structure, but a network of structures. It includes all of the endothelial cells of the capillaries throughout the entire brain. They are linked by tight junctions, so the only way that materials from in the blood can actually reach neurons and glia is if they are transported through those endothelial cells. This transport is carried out by transmembrane proteins that can bind nutrients like glucose or amino acids and actively transport them from one side of the cell to the other. This transport is regulated by the astrocytes, so they make up the second part of the blood-brain barrier. Anything that is lipid-soluble can get right across a cell. So oxygen, carbon dioxide, and steroid hormones do not need to be transported. Everything else must be recognized by a transport protein on those endothelial cells and astrocytes. That means anything that is not recognized by the human body and is not lipid-soluble should not get across the blood-brain barrier. For instance, antibiotic drugs and other chemicals. Some things can sneak their way across the blood-brain barrier and cause an infection of the brain called meningitis. Not every bacterium or virus can do this, but a few can do so from time to time. It turns out it's different ones that are more likely to cause meningitis under the age of 5 than over the age of 5. Typically, our immune system does a very good job of protecting us, so any form of immunosuppression can increase the risk of meningitis, as well as skipping the vaccines against HIV. Pregnancy and age can also be contributing factors. Let's review. Materials in the bloodstream cannot freely leave and get to neurons or glia. They must travel through one of two major pathways. At the choroid plexuses, a bunch of ependymal cells can take molecules from in the bloodstream and transport them, creating cerebrospinal fluid. This fluid is still not touching the brain. It's in the subarachnoid space and ventricles, so it needs to get through some more ependymal cells before it could actually reach a neuron. The second pathway, and there's a lot more surface area for this, is to be actively transported across endothelial cells. This means it's going across the blood-brain barrier. Furthermore, it might need to be transported through an astrocyte to reach much deeper neurons and glia. When waste products build up, these wastes must diffuse back across the ependymal cells to become part of cerebrospinal fluid, drain back into the dural sinuses at the arachnoid granulations, and this can now be brought back to the general circulatory system. So those are the structures that help to protect the brain. Next, we're going to learn about the major brain regions. Before we do, this slide here illustrates that even though we can discuss one brain region, many of the neurons in that region may make connections to very distant regions. Damaging this brain region may therefore have the same effects as damaging this region over here. Either way, I've lost an important connection. So keep in mind when we learn about these brain regions, you may see a lot of overlap. We may see the same role given to two different brain regions. And that's just because both are necessary for whatever that behavior is. So learning about the brain will be a lot messier than learning about any of the other organ systems that we've learned about so far, because of the high number of connections between these cells. With that in mind, let's begin talking about the major brain regions, starting from the most rostral location, the medulla oblongata. We'll talk about that, the pons, and the cerebellum, which is not shown on this picture, before we take a break. The medulla oblongata was our most primitive of brain regions, but it's also the most necessary. People do not get lesions to this area of the brain and survive. That is their cause of death. That's because the medulla oblongata controls our breathing, it controls our heart rate, blood pressure, and a number of other very important bodily functions. The pons helps the medulla oblongata, so these two brain regions work together. The cerebellum has a number of different functions. Remember, it's our second largest region of the brain, so we can't get through it all that quickly. It's involved in posture. It helps to fine-tune a number of our subconscious movements, and we're discovering these days that it's also responsible for some of our emotions. The neurons in the cerebellum make a huge number of connections with other neurons. This allows these neurons to carry out highly complicated computational tasks. This sort of math is necessary for basic behaviors like walking and simply standing upright. These behaviors actually require the coordination of dozens of muscles, with small changes made every millisecond, adjusted to inputs from the human body. As we can see over here in this slice through the cerebellum, all of the axons that connect these cells to other cells are bundled together in white matter called the arbor vitae. And superficial to that, you would find the gray matter, where all of the cell bodies and dendrites are located. Here are some close-up pictures of those dendrites, stained different colors. Over here we can see that there's a large amount of surface area for making those hundreds of thousands of connections. It's at this point in the lecture that I would have pulled up the game QWOP, which is a simple game where you try and get somebody to run, using only two muscles, the calf muscles and the quadriceps muscles, using the letters Q, W, O, and P to control those two muscles on the left and the right leg. To do that, you have to think about which muscles are being used as we move forward. And when we think consciously, we're using our cerebrum. This is the type of thing that our cerebellum does for us every day without having to think about it. Try out this game and see how good your cerebrum is at moving your legs in a way that gets the little runner to run forwards. And keep in mind that this is a highly simplified body. There's only 4 muscles to worry about, not 30 or 40. Damage to the cerebellum will cause a condition called ataxia, where the body cannot coordinate these sorts of movements. This can be caused by trauma or stroke, or temporarily by intoxication. The end result, though, is that muscle movements are not coordinated and cannot be mapped to visual inputs. For instance, touching your finger to your nose is a lot more difficult if you are suffering from ataxia. If you want to do a little homework, try intoxicating yourself and see how well you do on that sort of test. This young man here was born without a cerebellum, and when he was in the news, a number of doctors were quoted as saying they couldn't understand how he was even alive. Had they done a quick Google search, however, they might have noticed that there have been some other people born without a cerebellum. Nevertheless, based off of what we know, he should not be able to move his body at all, and yet he's learning to walk. That's a little difficult to explain based off of the basic science that we've learned, but look at this here. No pun intended. I find it very interesting that this young man is blind, and I find that interesting because it suggests a little bit of neural plasticity might be happening. Neural plasticity says that brain regions don't absolutely have to do the same job in every person. For instance, somebody who is blind may have a very heightened sense of hearing. This young man, because he is blind, that suggests a defect in his occipital lobe, which is present. It's his cerebellum that's absent, which may suggest that his occipital lobe is taking over for the role of the cerebellum. It's not very good at it. He is learning to walk, but will probably never walk like an average person. So that wraps up the medulla oblongata, the pons, and the cerebellum. Next up is the midbrain. finding the midbrain, it might be easier to identify the thalamus and then the pons and look in between the two. There are two major portions to the midbrain called the tectum and the tegmentum. More important for me is that you learn about the substantia nigra. This is a region of the midbrain that contains a lot of cells that produce melanin, the same type of stuff that we find in our skin, providing us with one of our three skin pigments. What is that melanin doing here in the brain? Obviously, no sunlight reaches here. And it turns out that melanin has more than one function. What we care about, though, is that when these cells are lost, as seen in the brainstem to the left here, this is what we find with patients who had Parkinson's disease. This disease causes a very specific motor defect. It causes both rigidity and tremors. Rigidity includes bradykinesia, the inability to begin new motions. If you were to give a Parkinson's patient a push and get them started walking in a forward line, they could continue walking forwards on their own. Their cerebellum can manage once the behavior begins. But if that patient were to run into an obstacle, they would stop and not be able to turn and walk in a different direction. That would be beginning a new motion. They also have trouble moving from time to time, known as rigidity, but then they also have problems with the exact opposite. They not only have problems moving, they have problems with too much motion. And this tells us a little bit about neurons. They are not really binary structures, meaning they aren't on or off. They're kind of constantly firing, and then we can increase the frequency or decrease the frequency. But when you take those action potentials away entirely, it leads to rather complex behaviors, such as rigidity and tremors, these opposing types of behaviors. Moving on, let's talk about the structures of the diencephalon, including the thalamus, the hypothalamus, and the pineal gland. The thalamus has a number of very important functions, but perhaps the most important is that it relays nearly all sensory information to the cortex. Our brain is receiving a huge amount of information all of the time from our various senses, but not all of that information is important. For instance, right now there might be a sound of a heater or an air conditioner going somewhere in your house. Because that sound hasn't been changing for the past several minutes, your thalamus probably decided that that information wasn't important, and it dead-ended here. It did not reach your cortex, where you would be consciously aware of it. Now that I've talked about that sound, however, your thalamus has maybe changed its mind and decided that that information really is important, and now you are aware of that sound. It's been there for however long you've been watching this lecture. You've just been tuning it out, and that's one thing that the thalamus can do. Decide what goes to the cortex to allow you to be consciously aware of it, and what does not. Below the thalamus is the hypothalamus, including a couple of structures that are easy to put on lab exams called the mammillary bodies. We also have the infundibulum, which connects to the pituitary gland. We covered the pituitary gland in BI 231, so I'm not going to discuss it here. The hypothalamus has a number of different functions, including the one that we talked about last quarter, regulating the pituitary gland. But the hypothalamus is also involved in our senses of hunger and thirst, and it also controls our base body temperature. The last tidbit of information for this portion of the lecture is in regard to the limbic system. And all I want to point out here is the limbic system refers to a group of different structures. Different books will have a different list of structures. Rather than try to pick which book I like best, I want to talk about some of those individual structures one at a time and talk about their functions. And the ones that I will discuss are the ones that are most important to medicine. Otherwise, I'm not going to be using the phrase limbic system. Nevertheless, what all of these structures should have in common is that they are subcortical, meaning they are subconscious. And so they typically regulate emotions, things that I could become aware of, but not things that I can control. That wraps up the midbrain and the diencephalon, and this represents a very good spot to take a break, grab a drink, and come back at a later time. Welcome back. In this part of the lecture, we're going to talk about brain regions involved in drug addiction. We'll also talk about memory, and this will be a long and complicated subject. For instance, why can't we decide what it is that we remember? Why is it that instead of easily remembering anatomy, you can remember jingles from commercials from 20 years ago? We'll talk about sleep and dreaming. We'll talk about why we need to sleep, what happens when we sleep, and we can even ask the questions, will we die if we were to stop sleeping for long enough? We'll also cover those gyruses and sulcuses and ask, why is the cortex all wrinkly? To do that, we'll be focusing on the cerebrum. We covered all of the other brain structures in the previous portion of the lecture. We'll talk about some very important bits of physiology, namely learning and memory and sleep, and how these two to one another. For review, we covered in the previous lecture the cerebellum, pons, and medulla oblongata. These were considered part of the hindbrain. We talked about the function of the substantia nigra. This was an important region of the midbrain. And the hypothalamus. These were part of the forebrain. The next hour, we're going to be spending on just the cerebrum. This is also part of the forebrain. The cerebrum is the largest part of the brain. We'll be covering different parts of the cerebrum one at a time, but what most of these have in common is that when they are functioning, we are conscious of some sort of behavior or perception. The cerebrum is divided into white matter and gray matter, with white matter typically being the deepest part. There are, however, some regions of gray matter called the basal ganglia that are completely surrounded by white matter. These basal ganglia are deeper structures that will have some functions that are subconscious. Everything that is happening in the gray matter, like we see over here, will represent activity that we are consciously aware of. Just keep in mind that the neurons that are located in the cortex, this outer gray matter, have their axons traveling through the white matter. The surface of the cerebrum is bumpy. Each one of these bumps we called a gyrus. Between two bumps is a little bit of a dip called a sulcus. There were also some rather large spaces called fissures. So the fissures separated the brain into different lobes, larger divisions than the individual gyruses. You may be asking what the function of the gyruses and sulcuses is, and the answer is to get more gray matter. Gray matter is where cell bodies and dendrites are located, and dendrites are where connections are made. To think deeper thoughts and to have more complicated ideas requires more connections, and therefore more gray matter. There are two ways to get more gray matter if we stick with the basic pattern of white matter on the inside and gray matter on the outside. The first way is to grow a bigger brain. The second way is to fold the brain. The problem with growing a bigger brain is this would require a bigger skull. and a bigger skull would make vaginal birth even more difficult than it already is. So this was not a viable option. Folding, on the other hand, allowed us to have more gray matter while retaining the same basic pattern of white matter on the inside and gray matter on the outside. This is what some people refer to as a kludge. Why not just grow thicker gray matter? That would have worked too, but that would have required a completely different set of instructions, rather than slightly changing the instructions we already had. White matter is composed of myelinated axons that connect one region of the brain to a very distant region of the brain. There are a couple of large tracts of white matter that get special names. The corpus callosum connects the left and right cerebral hemisphere, as does the anterior commissure. There's also a posterior commissure in there somewhere. Each of these tracts of white matter can be further subdivided based off of what they connect. These names I do not have on our lab list, and so I'm not going to cover them here either. So that's the basic organization of the cerebrum. Next up, let's talk about the basal ganglia. These are regions of gray matter completely surrounded by white matter. These deeper structures tend to be involved in subconscious activities, like emotions. There are a number of basal ganglia. Many of them are considered to be part of the limbic system that I talked about in the previous lecture. Rather than discuss these as a system, because they aren't all connected to one another, let's talk about these one at a time. Let's start with the hippocampus. This is a pair of structures that are deep inside the brain, and they're involved in what we're going to call long-term memory consolidation. This is not where memories are stored, but without the hippocampus, we can't store memories elsewhere. This part of the brain can be damaged in Parkinson's and Alzheimer's disease, which can cause anterograde amnesia. That I'll explain in a little bit more detail coming up. The function of the hippocampus is known because of, well, somebody had theirs removed. That's how we know how it functions. After patient HM had both of his hippocampuses removed to try and cure his epilepsy, he instantly had anterograde amnesia. He could remember everything leading up to his operation, but from the moment he woke up after his operation, he could no longer store long-term memories. He only had short-term memory, which meant 15 seconds later, he forgot whatever happened. This made it very difficult for him. He didn't remember the surgery after about 15 seconds. Every time he went to the hospital, he had to be reminded of what happened to him. What's interesting is that you could ask him what he did that day, and he would come up with an answer. He didn't say he didn't remember. He came up with a jumble of things from his past. Stuff that maybe had happened years ago or decades ago, pieced it all together into what seemed like a memory of that day. We'll talk more about the hippocampus when we get to learning and memory. For now, let's move on to the amygdala. These parts of the brain are responsible for fear conditioning and a few other functions. Fear conditioning is a type of learning. For instance, this rat down here may receive an electric shock, and if we play a sound every time the shock happens, the rat learns that when it hears the sound, if it jumps, it can avoid the shock. That's a very powerful way to learn, to try and avoid a noxious stimulus. Lesions to the amygdala can lead to a loss of fear, which is what we would expect. But because the amygdala is involved in other behaviors, we get some rather unexpected symptoms, like hypersexuality, hypoemotionality, and hyperorality. There are differences in the amygdala between patients with schizophrenia and those who do not have schizophrenia. That doesn't mean that the changes to the amygdala cause schizophrenia. It could be the exact opposite. Schizophrenia causes changes to the amygdala. These are not so significant that we can use this as a diagnostic basis. Alcoholism can damage the amygdala. If you've ever seen that guy at the bar who doesn't seem to be afraid of anybody and is willing to get into fights that he really shouldn't, you've seen this in action. Maybe you're aware of this down here, that oxytocin can suppress the function of the amygdala. Mothers experience much less fear when it comes to defending their children, and this would be suggestive of a function here of oxytocin. Next up are a pair of structures called the ventral tegmental area and the nucleus accumbens. These work together, but from here on out, let me just mention the nucleus accumbens. These are also involved in learning, the exact opposite of the way the amygdala is. These provide positive reinforcement. Activation of the nucleus accumbens results in pleasure, and it's involved not only in learning, but a special type of learning called addiction. This part of the brain can be directly stimulated by a number of drugs that we say are addictive, such as cocaine, amphetamines, nicotine, and opiates. A classic experiment with the nucleus accumbens was done on rats. These rats were trained to understand that they had a choice. They could press one lever for food or another lever to have opiates injected directly into their nucleus accumbens. But they could not do both. And these rats stimulated their nucleus accumbens to the point where they starved to death. Activation of the reward center is so powerful that it can overcome the pain and anguish of a slow death due to starvation. There was a problem with this experiment, and it was done on rats in cages. The experiment was repeated by a famous group from Canada a little bit later. And in this experiment, the rats were put into a rich environment. They were not isolated from other rats. They were in a group of rats, and they had toys and nice material to run around in and build nests out of, and they called this Rat Park, and then repeated the same experiment. Would rats starve themselves when given opiates? The rats would still administer themselves with opiates, but not nearly to the same extent as isolated rats. And one of the tantalizing conclusions from this experiment is one of the worst things that you could do to a drug addict is to isolate them in prison. That would only serve to reinforce their drug addiction, not to help them overcome it. We can't conclude too much from rat experiments. Nevertheless, that is the strongest conclusion that we can make. The nucleus accumbens is the brain region responsible for partner preference amongst these two different types of voles. Last quarter we discussed that oxytocin release could help the monogamous prairie voles to learn to mate for life. A lack of oxytocin receptors in the nucleus accumbens is what prevents the meadow voles from adopting a similar behavior. They are not capable of learning this as well, and therefore they do not mate for life, but instead are polygamous. The nucleus accumbens, therefore, is involved in learning due to positive reinforcement. This is what allows animals, such as humans, to learn to associate positive things with things that might be neutral, such as the sound of a bell or a tuning fork. This leads us to the topic of learning and memory. First, we need to divide memory into two different types. We have factual memory, or declarative memory, and we also have skill memory. Both of these require a lot of repetition, but there are some major differences between the two. Factual memory is more conscious, and we store facts in the context in which they are learned. For instance, In the Boy Scouts, I learned the difference between brown bears and black bears while on camping trips, but I can't really tell you the difference between those two right now while I'm sitting here in my living room. Apparently my brain just does not see that as relevant information for sitting in the living room. On the other hand, skill memory, sometimes called motor memory or even muscle memory, even though it's not stored in muscles, also requires repetition but is not stored with context. Meaning if you learned to ride your bicycle in Nebraska and then moved here to Oregon, you still know how to ride your bicycle. The creation and storage of factual memories begins in the prefrontal cortex. This is where short-term memory is located. To keep things in short-term memory requires constant activation of action potentials in some form of reverberating circuit. For instance, back in the day of remembering phone numbers long enough to write them down, we had to keep repeating the numbers over and over and over again. And if somebody interrupted you and asked you to solve a math equation, you'd probably forget that person's phone number that you just received. To store that number into long-term memory required activation of the hippocampus. In addition, help from the nucleus accumbens or the amygdala could aid the hippocampus in deciding what short-term memories should be consolidated or converted into long-term memories. Those long-term memories were stored someplace in the cortex, depending on what type of memory it was. Motor memory, or skill memory, involves different brain regions, including parts of the cortex and the cerebellum as well. I'll be focusing more heavily on factual memories, although many of these same things happen with motor memories. We start with a short-term memory. These can be recalled immediately, but can also be forgotten quite easily. And we're only capable of storing about 4 or 5 things in our short-term memory at any given time. If you learn a 6th thing, you'll forget the 1st. And if you learn a 7th, you'll forget the 2nd. This is why I advise you to break your studying up into small sections, rather than trying to cram everything into one large study session. You'll probably only be able to consolidate the last 4 or 5 things that you crammed into your head. To form a long-term memory involves converting those short-term memories into something more permanent, and we call this process consolidation. Our brain can store a heck of a lot of information. The amount of information is not really a problem. It's the organization. How do I recall the correct pieces of information rather than a bunch of garbage? And that's where context is important. We typically only recall things in the context in which we learned them. You're probably familiar with this. You spent a long time studying at home. You could remember everything when you quizzed yourself at home, and then you came to school to take a midterm, and your brain seemed to be blank. And then when you got home, it all came flooding back, and you got mad at yourself. Why did your brain do that to you? And that's because it was expecting context. It's going to remember that information when you're in the area in which you learned it. Luckily, you can trick your brain. Context doesn't always have to be the room that you're located in when you learned that information. It could be a melody, it could be a picture, it could be the color of ink that you use to write down that fact on a flashcard. There's a lot of things that could be considered context. Nevertheless, to store a memory requires more than one brain region. A memory is not a single location in the brain. It's a connection between a lot of different things. For instance, my memory of dogs might involve cold noses, fuzziness, warmth, and forelegs. So when we consolidate a memory, we're going to strengthen the connections between these concepts while weakening the connections with irrelevant concepts. That means that when we are born, we're already born with most of the connections already in place. In fact, we've got extra connections between neurons, most of which don't make any sense. And the process of learning does not involve growing new neurons or growing new connections, but strengthening the connections that are already there and removing connections that are nonsensical. To understand how these connections are strengthened, let's start by zooming in on a synapse here. Looking more closely, we can see that the axon terminal is capable of releasing neurotransmitter, which can bind to ligand-gated ion channels on the dendritic spine. If I release a little bit of neurotransmitter, this might cause a small graded potential, not one big enough to trigger a behavior. But with repeated stimulation, we could activate enzymes that could phosphorylate these ligand-gated ion channels and make them bind to the neurotransmitter more tightly. If this happens, releasing the same amount of neurotransmitter might cause a different response. I'll open up more of these ligand-gated ion channels. This will cause a graded potential, and maybe this will trigger a behavior. I will have changed, and we would say that I have learned something. Phosphorylation, however, is not permanent. This is not a short-term memory, but I could forget this by tomorrow or the next day as these phosphate groups pop off the ion channels. Luckily, this can start a string of events where with further repetition, the postsynaptic cell could increase the transcription of ligand-gated ion channels, and the presynaptic cell could make more neurotransmitter-filled vesicles. That way, when the presynaptic cell fires the same action potential, it's going to release more neurotransmitter, which can bind to more receptors, causing a big graded potential that might elicit a behavior. Protein expression is much longer term than phosphorylation. Still, this is something that I might forget in a month or two. It's not what we would call a permanent memory quite yet. But if I continue with the repetition, we will see even further changes. And to see those, we need to zoom out. Let's go back to our neuron connected to the next neuron. With a permanent memory, what we see are changes in neuronal structure. The presynaptic neuron can grow more axon terminals, and the postsynaptic neuron can grow more dendritic spines. That means we have more synapses between these two cells. Therefore, our connection is much stronger. When the first cell fires an action potential, I'm definitely releasing more neurotransmitter and activating more ligand-gated ion channels. These sorts of changes are what occur with permanent memories. We can see this happening under the microscope. We can watch dendritic spines growing as animals undergo learning. What will happen is that neurotransmitter release can not only activate action potentials, but can induce polymerization of the cytoskeleton, causing dendritic spines to grow towards the source of neurotransmitter release. Down here in the bottom left, you can see dendritic spines in two different animals, one of which has had her ovaries removed and treated with a placebo, and this one who's had her ovaries removed and treated with estrogen. We see that estrogen can increase dendritic spine growth, and we can also see that estrogen is beneficial for learning and memory. To summarize, a number of things can change in the brain as long-term memories are formed. The most rapid changes are changes in enzyme activity. We saw the phosphorylation of receptors which increased how long they remained open or how tightly they bound to a neurotransmitter. That's also the first thing to be reversed if repetition is not maintained. With further repetition, we can see more permanent changes, an increase in the gene transcription of ligand-gated ion channels or neurotransmitter-filled vesicles. Still, this is not what we would consider a permanent memory, as proteins do not last forever. A much more permanent memory involves structural changes to neurons, an increase in dendritic spines, and axon terminals can be visualized during permanent memory formation. All of these things are mediated through the activity of those neuromodulators that we discussed in Chapter 12. Remember that neuromodulators are chemicals that do not transmit signals, but change how those signals are received. These are the signals that lead to changes in gene transcription and changes in dendritic spine and axon terminal density. Glia can release these neuromodulators, and in fact we believe that it's the glia that decide which dendritic spines and axon terminals should grow and which ones should recede. This is why I said the glia were a lot more important than our textbook gave them credit for. The neurons can change their synaptic strength, but those changes seem to be guided by the activity of nearby glia, including astrocytes and oligodendrocytes. Here are a few more pictures for fun that show astrocytes coming very close to these synapses. And we believe that those astrocytes are regulating the strength of these synapses. This cartoon up here was drawn based off of this microscope picture down here. All of these things require repeated electrical stimulation, the release of chemicals called neuromodulators, and the activity of nearby glial cells. Keep in mind that we want to increase the strength of some of the synapses, but not all of the synapses. For instance, let's say we have a neuron down here that controls salivation. It's possible that a neuron that can detect the smell of food has a synapse that is strong enough to activate this salivation neuron on its own, but that activation of a neuron that detects the sound of a bell would not trigger salivation. With repeated stimulation, however, this can change. If both of these neurons fire at the same time, the salivation neuron can be triggered. The salivation neuron will in turn release a retrograde signal, a neuromodulator that will tell synapses to increase in strength. We don't want to increase the strength of all of the synapses, though. For instance, this neuron over here that fires an action potential when it hears the sound of the door, or this neuron over here that fires an action potential when we see a cat. So how could these synapses not change and only the sound of the bell change? And of course the difference between these is in activity. The bell and the food neurons are firing action potentials, and when they receive the retrograde signals, only those neurons will change their synaptic strength. The inactive neurons remain unchanged. Later, it may be just the sound of the bell will be enough to trigger the behavior of salivation. The sound of the door and the sight of the cat will still not trigger this behavior. That concurrent firing was also very important. The two neurons had to fire at the same time. For instance, you can't house train your dog by punishing your dog an hour after he's peed on the carpet. Positive reward at the same time is much, much more effective. Rewarding your dog for peeing outside is much better than trying to punish your dog later. In fact, if you come home and punish your dog for having peed on the carpet, the only thing your dog is going to learn is that he doesn't like it when you come home. That'll make him nervous and more likely to pee on the carpet. Our memories start off as short-term memories here in the prefrontal cortex, but then the hippocampus can decide which of these should be consolidated and stored elsewhere in the cortex. The hippocampus needs help from our emotional centers, like the amygdala and the nucleus accumbens. To repeat this in a different way, and I keep repeating myself because this is so important, the memories that get consolidated tend to be those that have emotional context, something pleasurable or something painful. So to store a long-term memory requires activity of the hippocampus. The memories aren't stored here, but this part of the brain is absolutely required. And it gets help from the amygdala and nucleus accumbens, the other basal ganglia responsible for fear and pleasure, respectively. The short-term memory gets a lot of information throughout the day. We don't consolidate all of that into long-term memories. Only those that the brain, the deeper parts of the brain, decide are important enough. You do not have conscious control over this, but luckily there are some things you can do to trick your brain into remembering information that you want it to. Try and give it some emotional context. Otherwise, it's not going to consolidate the boring A and P facts. It'll remember where and when somebody flirted with you, and where and when you ate a really good meal. So we say the nucleus accumbens and the amygdala are both essential to memory consolidation. Damage to either one of these can impair long-term memory storage. Complete removal of the hippocampus absolutely and completely damaged the ability to store any long-term memories. These memories, once again, are actually stored out in the cortex. Not in a single location, we have increased the strength of connections between neurons across broad regions of the cortex. That is why we can recall these memories consciously once they've been created, because they are out in the cortex. Where in the cortex they are located depends on what type of memory it is. Is it a visual memory, an auditory memory? Is it something you remember how it smells? Those will be stored in different parts of the cortex that we'll learn about here in a moment. Long-term memory consolidation requires repetition. The amount of repetition is going to depend on the intensity of the emotions going along with that memory. For instance, maybe you've suffered from food poisoning, or maybe you drank enough tequila to start vomiting. It may have only taken that one instance for you to learn that you're now going to avoid that food forever, or that you're never going to touch tequila again. It turns out that vomiting is one of the strongest emotional cues that we have. I don't suggest trying to use that to learn anatomy and physiology. It would be much better to try and give yourself positive rewards along the way. Nevertheless, learning anatomy requires some sort of emotional context, and that usually involves some sort of trick. A funny song, a silly joke, a dirty limerick, etc. Are there any drugs that you can take to increase memory performance? Turns out, yes, there are. Both caffeine and nicotine have been shown to boost memory performance. I would definitely not suggest taking up a nicotine habit just to do better in school. The benefits to memory are not that significant, and the side effects of a nicotine addiction are not worth the meager benefit of increased memory. Another class of drugs that can improve memory are called NMDA agonists. These can activate glutamate receptors. You're not going to see these drugs in use anywhere because glutamate is one of the most common neurotransmitters in the central nervous system. So these drugs would activate pretty much the entire brain all at once, not just memory centers in the hippocampus and elsewhere. What about more natural products? Well, nicotine is all natural, so we can't say that natural is better unless we say that nicotine and tobacco products are great. Ginkgo biloba is often touted as an all-natural improvement to memory, but the scientific evidence that we have is that it doesn't do anything of the sort. It certainly does not improve the deficits to memory that we see with Alzheimer's disease. On the other hand, blocking memories may be very important clinically. A class of drugs has been recently shown to do just this. You may ask, why would you want to block a memory? It turns out memory can be a very powerful mediator in drug addiction. Recovering drug addicts have a very hard time staying in recovery if they are around context that reminds them of drug use. For instance, the same location where they used drugs, or around the same people with whom they used drugs. If those people are their spouses or other family members, it's very difficult as a medical professional to tell that drug addict to stop seeing those people altogether. And yet, if they don't stop seeing those people, their chances of relapse are very high. This drug, which might be able to block memories, has the hope of increasing the rate that people recover from drug addiction without having to avoid context. Our previous treatments for drug addiction include drugs that simply antagonize the receptors that bind to those drugs. For instance, giving patients methadone, which can bind to opiate receptors more tightly than morphine or heroin. The methadone sits in the receptor and therefore attenuates any sort of high that that patient may get from the heroin that they used to use. This is not ideal because methadone does cause a little bit of a high. It's less addictive than heroin, that's for sure. It causes less health deficits than heroin, but it's still not 100% safe. Whereas the drug on the left here is a medicine used to treat blood pressure. This is much safer than methadone, so this shows great promise in the near future. Last up on the topic of learning and memory, there are different ways to learn fact-based information, and we call these ways different learning styles. This has led to all sorts of conclusions within the educational system. The first conclusion was that different people do better at different types of learning styles than others, and the evidence suggests that that's not really the case. In fact, what people should do is try out all sorts of different learning styles. If you rely on just one, you're not going to be able to learn as much as if you try all sorts of different learning styles. So that's what I would encourage you to do, not just rely on listening to these videos, but drawing pictures. And that involves moving your hands a bit. And read the textbook as well. Try using all of your different senses and all sorts of different styles, and you will probably maximize the amount of learning that you can do. So that was a rather lengthy tour of learning and memory. I focused most heavily on fact-based memory, but many of the same changes at a chemical and cellular level that occur with fact-based memory also occur with motor memory. Next up, let's talk about sleep. When we go to sleep, our brain activity does not decrease. It remains just as active as it is when we are awake. Its pattern of activity does change, however, and the pattern of activity can be measured by a device called an EEG. This device will detect electrical activity at the surface of the skin. It is not measuring action potentials, but the residual activity of millions of action potentials, and probably even more slow wave signals of the glia deep inside the brain, as that electricity leaks through the skull and skin to be detected by electrodes at the surface. This is not a direct measurement of anything. It is very indirect. Nevertheless, we can glean some very useful patterns from it. Those patterns include brain waves. And it turns out that brain waves change from when we are awake to when we are asleep. We can divide sleep a number of different ways, and I would like to keep it fairly basic. Based off of EEG patterns, our brain activity when we are awake changes when we sleep. And when we sleep, we go through two basic phases of sleep. We descend into a deep sleep pattern, and then back into REM sleep. And then we alternate between deep sleep and REM sleep until we eventually wake up once again. The EEG pattern during deep sleep is a lot slower, whereas during REM sleep, the frequency of the stimuli is a lot higher. During REM sleep, furthermore, our eyes move rapidly. This is where it gets its name for rapid eye movement sleep. During deep sleep, our eyes do not have this motor pattern. It is sometimes said that we dream during REM sleep, and it turns out we dream during both REM sleep and deep sleep. It's just that the dreams are different, and we're more likely to remember the dreams that occur during REM sleep, hence the misunderstanding that dreams only happen during REM sleep. During deep sleep, the brain tends to replay some of the motor activities that it had undergone over the past few days. This is pretty boring, hence you are less likely to remember this when you wake up. Nevertheless, if you were to try and train somebody in a new skill, and then you woke them up that night every time they entered into deep sleep, but you allowed them to undergo REM sleep, they would have great difficulty in learning that skill. On the other hand, during REM sleep, we tend to replay factual memories. This is where consolidation of factual memories occurs. If you were to teach somebody a whole bunch of facts, and then when they went to sleep that night, wake them up every time they entered REM sleep, but allowed them to go through their deep sleep cycles, they would have greater difficulty recalling those facts in the morning. So, sleep is the time where we do most of our memory consolidation. And we have two basic types of sleep, one for our motor memories and one for our factual memories. It's during sleep that the protein synthesis required for long-term memory consolidation occurs. This is when we make more of the ligand-gated ion channels and make more vesicles filled with neurotransmitter. This is when we would grow new dendritic spines and axon terminals. Sleep loss, therefore, is very significant and in fact contributes to a large number of automobile accidents. This is comparable, for sure, to automobile accidents caused by drunk driving. Sleep loss was also involved in a number of major disasters, like the Exxon Valdez oil spill, the Chernobyl and 3 Mile Island meltdowns. I have heard stories that people will die if they do not get enough sleep, and this is not true. It would be true if you were a rat, but as a person, it's not going to kill you, unless of course it leads you to die in an automobile accident, or to allow the nuclear reactor where you work to melt down while you're still inside the building. The reticular activating system is a brain structure that's very important to sleep. We call it a system because it extends throughout the entire brain, but you can think of it as a gate. It blocks signals from going up or down as we sleep. This is very important. For instance, it prevents sensory information from reaching our brain as we are sleeping. It also prevents information from leaving the brain as we sleep. And this is important because the same neurons that were active during the day are also active when we sleep. If we're dreaming about running, the same neurons that actually cause us to run would be firing action potentials as we dream about running. And if it weren't for the reticular activating system blocking the action potentials from leaving the brain, it would cause us to actually move our legs as we dreamed. People who have a deficit in the RAS may suffer from conditions like sleepwalking or other movements that occur during sleep. So, it's very important to get enough rest. This is when memories are consolidated. And if you want to do well in this class, I know it's easy for me to say, but it means getting a good night's sleep. It's been estimated that 1/4 of us here in the United States suffer from some sort of sleep disorder. But that's very hard to quantify because different people need different amounts of sleep. It's estimated that an adult needs about 8 hours of sleep per night. If you're somebody who's selling mattresses, you may quote a number that's even higher than that, but that's not based on any good scientific evidence. However, recently, some better evidence has come out to suggest that 8 hours is probably on the upper end. People actually live the longest if they get, on average, 6 to 7 hours of sleep per night. Still, different people are different, so we can't judge too much based off of these numbers. So that wraps up sleep, and this would be a very good spot to take a break, maybe get some sleep, and pick up on the 3rd section of the lecture tomorrow. Welcome back. In this 3rd of 3 parts, we'll talk about Phineas Gage, the man who suffered an accident building a railroad and lost a part of his brain and became a big jerk. Or at least that's the story you've probably been taught in other classes. There's a problem though. It skips over a lot of information and it's a very unscientific way of trying to learn about the human brain. So I'll clear up a few things about that. We'll talk about decision making and why sometimes our brain makes very bad decisions. We'll talk about phantom limb today, and this will relate to sensory homunculi. And it'll also illustrate how neurons aren't like on and off switches. We'll talk about left-brained and right-brained activities, and while it's definitely true, there are things that the left side of our cerebrum take care of and things that the right side of the cerebrum take care of. Nobody is left-brained or right-brained. We all use both sides of our brain. We'll talk about serotonin theory of depression once again, and I will introduce to you a better theory. We'll talk about the growth of neurons. We'll also discuss grain brain, is sugar toxic to the brain, and we'll also talk a little bit about what we're learning about autism. To do all of this, we'll start off with the prefrontal cortex and discuss Phineas Gage, what we can learn from him, and learn a bit about decision making. We'll talk about hemispheric lateralization. What this means is that each hemisphere of the cerebrum is different on the left side and the right side. We'll see that there are different tasks that the two sides of the brain take care of. And lastly, we'll talk about some of the common disorders of the central nervous system, focusing on what we've already learned about. We'll talk about Alzheimer's disease, we'll talk about depression and what this tells us about the growth of neurons, and we can talk a bit about stroke. We're going to start today by learning about the prefrontal cortex. And whenever we talk about the prefrontal cortex, the name Phineas Gage pops up. He was a railroad worker in the 1800s who was tamping down on a stick of dynamite with a metal rod when the dynamite went off, blasting the rod through his skull and taking out a large chunk of his prefrontal cortex. Afterwards, as stories go, he became a big jerk. Now this sounds exactly like the way that we've learned about the brain. Remove a part of it and ask what changes. Unfortunately here, we do not have any first-hand accounts of what actually happened to Phineas Gage. We have no reason to doubt that he lost a chunk of his brain, but we don't have any first-hand accounts of how his personality actually changed. And what we do know is that at this time, at his pay scale, the most likely form of painkiller that he would have access to was alcohol. And I would imagine it would take a lot of alcohol to numb the pain from having a spike shot through your skull. So the question is, did he become a big fat jerk because he was missing a part of his brain, or because he was self-medicating with a lot of alcohol? Certainly, self-medicating with alcohol can make people jerks too. So this makes his story a little bit messy. But even messier, and this is always overlooked in textbooks, is that he got better. He did lose his job working for the railroad, but later he came back to it and ultimately got a pension from the railroad company. Now that leads us to two possibilities. One, he grew that part of his brain back and stopped being a jerk. Or two, his wounds healed over enough that he no longer needed to medicate with as much alcohol as he did, and his personality returned to that where he could hold down a job. And it seems much more likely to me that he quit drinking so much than he grew a chunk of his brain back. So he's a very interesting story, and he can teach us a little bit about science, but I think he's a better story about teaching us about the scientific method. We shouldn't rely too much on stories, especially secondhand stories, and especially stories that have been crafted by a doctor that toured with him around the United States, showing him off to other medical schools as sort of a medical curiosity. And we should cover all of the facts that we know, rather than try and jump to easy conclusions. The story was partially right, if not for the correct reasons. The prefrontal cortex is involved in decision-making, and we could say that's an important part of our personality, but it's definitely not the only thing about our personality. In the previous section, we talked about the amygdala and nucleus accumbens. Certainly our fears and our desires are part of our personality as well. But making decisions is a very important part of social behavior modification. For instance, do we just take the things that we want, or do we respect other people's property? Do we think ahead to say that, oh, if I were to steal that, I could get in trouble? That's the type of thing that the prefrontal cortex seems to be able to do, to weigh the pros and cons and come up with decisions. The prefrontal cortex is one of the last regions of the brain to mature. After we are born, there are two periods where the brain grows significantly, with lots of new neurons created. Many of these neurons make connections that are not useful to us, and so these connections, and in fact entire neurons, must be pruned and removed. This is guided by glia, and once they are sure that the pruning has been done properly, what neurons are left are myelinated, which pretty much cements them in place, and will no longer be significantly changed throughout the rest of our life. The prefrontal cortex is the last region to do this, and doesn't finish with myelination until our 20s. When the neurons in the brain are unmyelinated, they are capable of a lot more plasticity. They can make very different connections if need be. That's why it's easier for us to learn a foreign language when we're children than it is as adults. When we're adults, we can really only change the dendritic spines and axon terminals. We can't make any major structural changes because the neurons have been cemented in place by the myelin. Damage to the prefrontal cortex can occur with a concussion or a traumatic brain injury. Back when I was a kid, we didn't think too much about concussions. In fact, if you could count the number of fingers on the coach's hand, you were sent back in to keep playing whatever sport it was you were playing. But these days, we now realize that a traumatic brain injury can have very long, lingering effects on the brain, and we're still learning what those effects are. This has led to changes in how kids play sports and how coaches at high schools and other schools are trained differently. And this may also be changing how the NFL operates. That's been in the news quite a bit this year. Now it's time for me to plug my journal club. Schizophrenia can be caused by damage to the prefrontal cortex. This damage occurs in 2 steps. The first step occurs during fetal development and can be caused by a deficiency in growth factor signaling. This leads to too few neurons growing in the prefrontal cortex. This damage can be uncovered later in life when the prefrontal cortex is maturing. Prior to myelination, unneeded or unuseful neurons need to be pruned. With excessive pruning, this could unmask the deficiency in growth that occurred during fetal development and lead to the first symptoms of schizophrenia. If you wish to know more, follow the link I have there at the bottom right. One of the main tasks of the prefrontal cortex is in decision making. And it appears that there are 2 ways that the prefrontal cortex can make decisions. It can either do so very quickly, sometimes this is called a reflexive decision, or it can do so slowly, make a more conscious or reflective decision. It's good to be able to do both. Sometimes we just like to come up with answers quickly, especially if time is of the essence. So reflexive decisions are very quick and don't involve a large number of synapses. On the other hand, more conscious decisions take attention and effort. This may affect you. For instance, if you were to tell a patient that their surgery has a 90% success rate, they're going to be more likely to think that's a good idea than if you told your patient their surgery has a 10% failure rate. It's the same information, it's the same data, but how you phrase it is going to influence whether your patients opt to do that surgery or opt not to. Why does our brain have 2 ways of making decisions? Well, if you were to see this guy in a dark alley, you probably wouldn't want to spend a long time deciding whether or not he was going to be your new best friend. On the other hand, there are certain things that we just can't come to very quick snap decisions. For instance, why is this comic here super darn funny? It's going to take you some time to read it, and you may even have to think about what you've learned in this class. Let's go through an example. I have a math problem for you. We've got a baseball and a bat that together costs $1.10. And the bat costs $1 more than the ball, so how much does the ball cost? And many of you are probably thinking, that's easy, it's 10 cents. There's a problem with that though. The bat costs $1 more than the ball. So if the ball costs 10 cents, the bat would cost $1.10. And we said that together, the cost of both of these was $1.10. So that can't be the answer. Hopefully you now realize that the correct answer was actually 5 cents. That's took me a while to get to the correct answer, and I didn't realize I was wrong at first until it was pointed out to me. My brain had made a reflexive decision. It saw 2 numbers, and then it just did some quick math on them, came up with a difference, and I said 10 cents. And I would not have thought about it anymore had somebody not pointed out to me that I needed to. But once it was pointed out to me, I made a more conscious decision. I did some algebra on this and came up with the correct answer. So this just illustrates that our brain will tend to rely on these reflexive, quick decisions, unless it has some reason to spend more effort on that sort of decision. Here's another fun example of the function of the prefrontal cortex. Count the number of times the players wearing white pass the basketball. Okay, good. So maybe you've seen this video before and got the correct answer 15 passes. When I show this in class, about half of the students miss the gorilla. So let's just review the video. And sure enough, somebody in a gorilla suit walked halfway through and beat his hands on his chest. How could half of the students in a classroom not see that? And this just illustrates that the prefrontal cortex can only deal with about 4 or 5 pieces of information at a time. And if it gets overloaded, we just miss everything else. So you were trying to pay attention to the players in white passing the basketball while ignoring the players in black, and that took up all of the space in your prefrontal cortex. It had to make a reflexive decision about whether the vision of the gorilla was important enough to send to your cortex to be aware of it, and you weren't. The rest of the cortex has the same basic organization as the spinal cord, in that motor information tends to be more ventral, and sensory information tends to be more dorsal. There are 2 major regions where this is very important. The pre-central gyrus is our primary motor cortex, meaning that when this part of the brain is active, it is controlling a part of the body. Just behind that is the primary sensory cortex. That means when this part of the brain is active, a part of our body is registering some sort of sensation, like touch or pain. These and a number of other regions of the brain were mapped out by a man named Dr. Wilder Penfield. He was a neurosurgeon who worked on patients with epilepsy. He couldn't see where the problem was in their brain. He just had to find it. And in order to do that, the patients had to be awake while he was performing surgery. He could stimulate different parts of their brain until he found the place that triggered an epileptic seizure and then remove it. But what he noticed is that when he stimulated other parts of the brain, there were certain regions that would cause the patient to move a part of their body. That was the precentral gyrus. And when he stimulated the postcentral gyrus, patients reported feeling sensations in those parts of the body. And this was very consistent. So he came up with maps of the cortex that we still learn about today. These maps can be further subdivided into specific regions, and we call these maps a homunculus, or a little person who lives inside your brain. These maps are not one-to-one. Some parts of our body take up a lot more brain space than other parts. And that's because I've got a lot more neurons in highly sensitive areas in my body than I do in areas that are a lot less sensitive. For instance, my fingertips are a lot more sensitive than my elbows. Hence, my elbows take up a lot less space in the brain. It takes fewer neurons to control the muscles around my elbow than it does to control the muscles in my fingers. Here's our textbook picture of what these maps look like. And you can see that regions of the body that are highly sensitive, or regions of the body that we have a lot of control over, take up a lot more brain space than other regions. Our lips, our hands and feet take up a lot of space, wrists and elbows a lot less. One major difference between these 2 maps are the teeth. There we go, over here. Those are highly sensitive, but I can't move them. Same with the genitalia, highly sensitive, therefore they take up a lot of space on the primary sensory cortex, but we don't move them, so they don't have any space in the primary motor cortex. 60 years went by before anybody mapped out whether female genitalia mapped to the same region of the brain as male genitalia. And it was discovered that females were a little bit more complicated than men. Maybe you didn't need to come to this class to learn that. What's interesting is that stimulation of the nipples could cause 2 different responses. It could cause a more functional response, activating a part of the brain associated with the chest area, or it could cause a more arousing response, stimulating a part of the brain that had overlap with the genitalia. Phantom limb also relates to these homunculi. I have a link to a very good TED Talk down below, or you can read that man's book. It's fascinating, but I'll summarize it quickly. If somebody loses a limb, now there's a region of their brain no longer connected to a part of the body. And what should happen is this part of the primary sensory cortex should take a permanent vacation, but it doesn't. And what might happen is that axons connected to nearby regions invade this now naked region and make new connections. Hence, sensory information coming from the face can now stimulate this part of the brain, which makes somebody think that their arm is being touched as well. The post-central gyrus we called our primary sensory cortex, meaning this is where we detect touch and pressure or other sensations from the skin. Our other senses are detected in other regions of the cortex. For instance, vision is sent back here to the occipital lobe, hearing to the temporal lobes, taste and smell are a little bit deeper inside here in regions called the insula. In between all of these are what are called association areas. As you are watching this video, your eyes are sending information to your occipital lobe, and your ears are sending information to your temporal lobe, and it's in between these two that those two bits of sensory information are combined or associated with one another. We do this for motor information up here, visual and other sensory information back here. Up until this point, the only difference between the left and right sides of the brain has been that the left side of the brain is connected to the right side of the body, and the right side of the brain is connected to the left side of the body. But there are some other regions of the cortex where this is not true, and certain tasks are only taken care of on the left side or the right side. And we call this hemispheric lateralization. Keep in mind that what I'm about to teach you is true for the average male. Not everybody will have the same maps of the brain. They may be flip-flopped, for instance. But it'll still be true that certain tasks are carried out by one side of their brain and other tasks by the other side, even if those sides are different from the average male. The left side of the brain frequently takes care of reading, writing, and math-based tasks, whereas the right side of the brain is more interested in spatial relationships and facial recognition. The ancient senses, vision and hearing and whatnot, have distinct regions of the brain that deal with them. Vision, for instance, is strictly taken care of by the occipital lobe, and it does a very good job of that. Human language, however, is much more recent, and our brain does not have a single area to deal with language. Instead, it's cobbled together a number of different spots. For that reason, I do not recommend relying on reading comprehension alone. Try and get as much visual and hearing information in as possible. Wernicke's area is one of these language areas. It's more dorsal, so it's one of the more sensory-associated language areas. Much of the time, it's on the left-hand side, and this part of the brain deals with understanding written and spoken language. If somebody were to have a stroke and damaged Wernicke's area, they could speak words just fine, but they could not put them together into sentences that made any sense. Broca's area also deals with language. This one is more ventral, and hence it is more associated with motor activity. Damage to this part of the brain leads people to not be able to say words properly, and they often get very frustrated because they can think in sentences just fine, but the muscles of their mouth do not respond properly and won't make the sounds that they want them to. While the left-hand side of the brain is dealing with language tasks, the right-hand side of the brain is dealing with spatial tasks, such as drawing. Because most of us have a functional corpus callosum, these two sides of the brain can communicate with each other and work together. But for a few people who've had their corpus callosum cut in order to relieve epileptic seizures, a very interesting phenomenon can be seen. If you show a picture to these people up close, and that picture is of a pirate who has an earring in one ear, this earring is only detected by one side of the brain, not the other. If that earring is detected by the left side of the brain, you can ask them, "What do you see?" And they will tell you, "I see a pirate with an earring in his left ear." On the other hand, if that pirate has an earring over in this ear, only this side of the brain would register that earring. Now, if you ask that patient what do they see, they will tell you a pirate. And if you ask them, does that pirate have an earring, they would say no. But if you ask them to draw what they see, they would draw a pirate with an earring. This side of the brain is responsible for drawing that pirate. And again, you could ask them, why did you draw a pirate with an earring? And they would say, I don't know. So we had to do this by showing them a picture up close. Otherwise, for pictures at a distance, both eyes would see the earring. So these patients never really notice any sort of deficit until they're put into a very artificial situation. Again, for the rest of us who have a functional corpus callosum, if this side of the brain over here sees the earring, it can tell this side of the brain that there's an earring, and then the patient could talk about that earring just fine. It used to be thought that some people were left brain dominant and other people right brain dominant, and that's just not true. What is true is that the two hemispheres carry out different tasks. The left side typically deals with reading, writing, and math, whereas the right side deals more with spatialization and facial recognition. However, with more modern imaging techniques, we have learned that everybody uses 100% of their brain. Nobody is left-brained or right-brained dominant. But maybe you've heard that we only use 10% of our brain, and that's just not true. We use 100% of our brain, just not all at once. Unless, of course, you have epilepsy. An epileptic seizure would be an instance where somebody was using 100% of their neurons all at the same time. So where did that number come from? Well, it came from a man who was selling a book trying to convince people that there were untapped powers in their brain, and that if they just gave him some money, he could teach them how to gain psychic abilities. There was never any science behind that, and yet it has been passed off as a scientific fact now for decades within our culture. That wraps up the basic regions of the cerebrum, including a number of important basal ganglia, and then we talked about the cortex areas today, motor and sensory cortex, the other sensory cortex areas, and then hemispheric lateralization. Before we move on, let me just recap a little bit about what we've learned and try to put it in some perspective. Let's start with the number of synapses that we have in our brain. If every neuron makes about 5,000 synapses, and we've got 100 million neurons, that number's so large it really doesn't have any sort of meaning to me. Maybe this is a better way to put it. If everybody on the face of the earth were to create a Facebook account and make 8,000 friends, that would be the same number of connections on Facebook as we have connections between the neurons of our brain. That, of course, ignores the glia, which also make a lot of connections, and we've got a lot more glia. How many connections do they make, and how many more glia do I have? Well, that we're still not really sure about. Our brain is a relatively small organ. It only takes up 2% of our body weight, but it uses 10 times that amount of our energy. It takes a lot of ATP to keep this sort of electrical field going. That's roughly the equivalent of 1/20th of a AA battery. So we've got a lot of battery power up there in our skull. Does that make our brain more powerful than a supercomputer? Well, that depends on your definition, I suppose, of powerful. For instance, I could go to the dollar store and for $1 or less buy a piece of plastic and circuitry that would give me a more accurate number for what is the square root of 2 than your brain can. Does that make your brain less powerful than $1 worth of plastic and circuits? Well, in some ways, yes, it does. But in other ways, no. Computers are really bad at determining that this is a picture of a dog and this is a picture of the same dog. Why are computers bad at that? Well, that's because computers work with circuits that are binary. They're on or off. And it's true that action potentials are all or nothing, but when that action potential reaches the axon terminal, it triggers the release of a number of neurotransmitter-filled vesicles. How many? Well, it's an average number. Sometimes it'll be a little more, sometimes it'll be a little less. And that means our brain is better at dealing with fuzziness than a supercomputer is. We are able to tell that this is literally not the same photograph, but the fuzziness of our logic is able to accept that these could be the same animal. Action potentials travel at 200 miles an hour. Computer signals travel a lot faster than that, which is great for a computer, but obviously we don't need faster signals. Works pretty well for us. If you were a flatworm, your brain would have 302 neurons, and they would be connected the exact same way as your buddy, the other flatworm over there. And both of you, it would have taken 9,000 genes worth of instructions to program that brain how to make those connections. Humans have a lot more neurons, and yet we've got roughly the same number of genes. That means the neurons in our brain aren't guided by literal instructions as to where to make connections. We seem to make connections randomly, and then later learn whether those connections make sense or not. And when they don't, we prune them. We get rid of those connections. But it takes us time to learn that stuff. And for years, we're pretty much useless, not capable of feeding ourselves or moving around or hunting or anything. Whereas these flatworms are pretty much ready to go. They are, however, exactly like every other flatworm when it comes to their nervous system. Whereas we humans have a lot more variability in our brains, and that leads to a lot more adaptability in our species. Lastly, one of my favorite analogies of the brain came from this book, and that points out that we've got this very ancient hindbrain. It's very similar to the hindbrain of lizards or other animals. We've got this midbrain that's still similar to the midbrains of birds, but what makes us unique as humans is our cortex. That's very different from most other animals, and evolutionarily speaking, is a lot more recent. It's the new part of our brain. And yet we still got these old parts of the brain that are still functional. They all have to work together. So the metaphor here is, imagine building a Formula One race car, this fancy new race car that had a functional Model T in the center. Not just a Model T, but a Model T that was still working, and your race car had to rely on that Model T for certain parts of the race. That is our brain. Sometimes your cortex gets really mad that the rest of your brain, like your hippocampus, somewhere deeper, decided not to remember some of the anatomy facts. Let's move on to discuss some common diseases that affect the brain and ask, how can we understand these better based off of the physiology that we just learned over the past hour? Let's start with Alzheimer's disease, which typically affects the elderly and causes damage to wide regions of the brain. This can lead to confusion, memory loss, and mood swings. That's going to be very relevant to you because your patients might behave violently towards you from time to time. We don't really know what causes Alzheimer's disease. What we do know is that people who die from Alzheimer's disease have amyloid beta plaques in their brain and phospho-tau tangles in their brain. and both of these seem to be associated with damage to the brain. There's a problem though. These might not cause Alzheimer's disease. They're correlated with it, that's for sure, but it could be that Alzheimer's disease causes these things to show up. Either way, A-beta and phospho-tau kind of seem like cellular garbage. And when that garbage piles up, it may cause damage to regions of the brain, poisoning neurons, causing them to die. And so we're now starting to ask, why does that garbage pile up? Shouldn't it be removed by the microglia? And sure enough, microglia are definitely associated with Alzheimer's disease. There's a large amount of evidence showing that microglia can protect against Alzheimer's disease when they are functioning properly. On the other hand, sometimes when garbage builds up in the brain, those microglia go overboard, and after they're done clearing out the garbage, they clear out neighboring neurons. And so altered microglial function is also associated with a worsened prognosis with Alzheimer's disease. So which is it? Do microglia protect us or hurt us? And the answer is, well, it seems they can do both. There are other neurodegenerative diseases like Huntington's and Parkinson's disease. These affect more specific regions of the brain, like the basal ganglia or the midbrain, substantia nigra. Let's return to depression and talk about it in more detail. One of the major theories about depression is that it's caused by not enough serotonin. Or wait, maybe it was caused by too much serotonin. Nevertheless, we know that this class of drugs, the SSRIs, are very successful at treating many patients, but not all, who have a form of depression. Let's contrast this drug with LSD. LSD mimics serotonin, and it combined to serotonin receptors found throughout the brain, causing many different symptoms, including visual hallucinations. SSRIs, on the other hand, inhibit the removal of serotonin from synapses, and this increases serotonin concentration in those synapses. So why do these two types of drugs work so differently if they both increase serotonin signaling? That has to do with whether synapses are active at the time. If I have 4 synapses, but only one of them was releasing serotonin, if I added an SSRI, it would only affect the synapse that was already active at that time. It would increase the amount of serotonin at that synapse, leading to a result, facilitation of the postsynaptic neuron. LSD, on the other hand, because it mimics serotonin, would activate all serotonin receptors at the same time, activating a lot of neurons in the brain, leading to effects that are not very specific or targeted. For this reason, medications frequently inhibit things that we want to have happen in the brain. So do we want more serotonin signaling, or do we want less serotonin signaling when we are trying to treat depression? Depression is not a single disease. It's actually a large number of diseases, and that makes it more difficult to treat. Some of the earliest drugs for treating depression affected not only serotonin levels, but also dopamine and norepinephrine. Nevertheless, with newer drugs being invented, we ultimately landed on this serotonin theory of depression, based on the fact that we knew some drugs worked, and that's what they did in the brain, increased serotonin signaling. Of course, those drugs don't work right away. SSRIs can boost serotonin levels immediately, but they don't have effects on depression for months. So this led to the alternate theory that maybe depression was caused by too much serotonin, and these drugs work by inducing a negative feedback loop, making the body less sensitive to serotonin. Still, both of these theories have some problems. What we have never seen in patients with depression is too much or too little serotonin in any part of their brain. What we have seen, however, is that stress causes the release of glucocorticoids, which can lead to the loss of neurons in brain regions like the hippocampus, and this can be associated with depression. And if the death of neurons might be important in the development of depression, the growth of neurons is also important. It was recently discovered that in Amish families, a type of depression could be passed on from generation to generation. And in these same families, a type of dwarfism could also be passed on. But there were no instances in medical records of somebody with this type of dwarfism also having this type of depression. It seemed that a mutation in the protein sonic hedgehog protected these people from depression, even if it caused dwarfism. This protein is responsible for the growth of numerous parts of the body, including certain regions of the brain, and changing how that region of the brain grows seems to protect against depression. The newer theory of what causes depression is called the neurogenesis theory, and this states that depression is caused by a lack of neurons in specific brain regions like the hippocampus. And there's two main ways that this can happen. Either people do not grow enough neurons in the first place during prenatal development. The growth of these neurons is under the control of growth factor molecules like sonic hedgehog. Secondly, people may lose too many of these neurons later in life, especially if they are exposed to chronic levels of the stress hormone cortisol. If these neurons that are lost release serotonin, then it's understandable that drugs that can boost serotonin signaling might be able to treat depression. But these drugs would only be masking the symptoms, not getting at the underlying cause of the disease. So hopefully in the future, we will have better treatments that actually get at the cause of depression. In the meantime, the best that we have seem to be able to manage symptoms for many patients. Before I move on, I want to point out that while there is a link between depression and suicide, the link is very complicated. In fact, many SSRIs, even though they are capable of treating depression, actually increase the risk of suicide. If you have a patient showing up in the emergency department who has just attempted suicide, you should definitely not give them an SSRI. There are other drugs that can lower their risk of going through with the suicidal ideations, including the club drug ketamine, which is also a horse tranquilizer, but makes people very, very happy and makes them much less likely to commit suicide. We do not give this drug for long-term treatments. This is just a short-term stopgap mechanism, but it's much more successful at preventing suicide attempts than SSRIs. As the nervous system gets older, we lose cells. And because most neurons do not divide, it's hard to get them back. And so we see a decline in neural function as we get older, and that's just part of the aging process. But some things can speed this up, like Alzheimer's disease, or other things that can cause senility. One of the most common causes of damage to the central nervous system is arteriosclerosis, which is a hardening or narrowing of the blood vessels, which can restrict blood flow to certain parts of the brain. This could cause a slow buildup of damage over time, or if that artery completely occludes or even ruptures, that could cause a stroke or a cerebrovascular accident, causing a massive amount of damage all at once. On the other hand, can we protect our brain from this sort of damage over time? And the answer is yes, we can. Exercise can do this, either mental exercise or physical exercise. Learning induces the growth of connections between neurons. And if we have more connections, we can afford to lose a few without seeing any sort of deficits. So doing crossword puzzles or physical exercise, both of these things have been shown to reduce the effects of Alzheimer's disease. It may not prevent Alzheimer's disease, but if people get it, it's a lot less worse, and we start seeing the symptoms of Alzheimer's disease later rather than sooner. Just be careful what you promise. If you're going to promise to prevent Alzheimer's disease in your patients, you better make sure that you have evidence for that directly. This company here got sued for selling something that was based off of other people's research, but wasn't necessarily the same research. What about diets? Can eating the right food protect the brain? Well, if you have certain forms of epilepsy, then a low-carb or a no-carb diet can absolutely be beneficial. And this has been known for over 100 years. But what about for healthy people who do not have epilepsy? And in that case, there's no evidence to suggest that low-carb diets protect the brain and prevent diseases like Alzheimer's disease. And in fact, scientific evidence would suggest that maybe a low-carb diet is not that healthy, because the brain relies on very simple carbohydrates for energy. It doesn't like burning fat. It prefers sugar or sugar equivalents like acetic acid. That doesn't mean that these diets are bad for your health. It just means we don't have any evidence that they are good for your brain. Another disorder of the nervous system that we can understand in better detail now is spina bifida. This is a condition where the spinal cord is exposed and the interior portion of the spinal cord is open to the outside world. This is very dangerous because it could lead to meningitis very easily. And now we can understand this better because we understand that the nervous system started off as part of our skin and ultimately folded up into a neural tube. Spina bifida is a condition where that folding didn't complete, and part of the spine is still in this unfolded state. And this needs to be fixed very quickly by a surgery. We're starting to understand a little bit more about autism. What we understand very clearly is that Andrew Wakefield is a big fat jerk, and he lied when he claimed that autism was caused by the MMR vaccine. He owned a patent on a competing vaccine and could only have made money if he discredited the one that was used commonly in the United States and in Great Britain, and that's exactly what he did. Nevertheless, people still think that autism is caused by vaccines. Even our president has said as much. We, in science, know this is definitely not the case. When thimerosal, the mercury compound that people thought was causing autism, was removed from vaccines, autism rates have continued to go up. When you look at children who are unvaccinated, they have autism at the exact same rates as children who are vaccinated. So we are absolutely certain that autism is not caused by vaccination. So what does cause autism? Well, we'd probably have a much better understanding of what causes this spectrum of diseases if we hadn't been wasting so much time over the past 20 years trying to chase down this vaccination ghost. Still, we're starting to learn a little bit about what can put people on the spectrum. One risk factor is fathers over the age of 40. And another risk factor might be more environmental. For instance, mothers who have low levels of carnitine in their prenatal diet are more likely to have children on the autism spectrum. So that wraps up the neurogenesis theory of depression. We'll be talking a bit more about cell division in neurons when we get to chapter 14. We also talked about how the central nervous system changes with age, especially with Alzheimer's disease. And we talked just a little bit about the autism spectrum. But that wraps up this chapter.